Good afternoon, students. Now, today we are going to talk about the development of herd mania. So, yesterday we have already completed the other systems like circulatory system, respiratory system, digestive system. And now the last topic in this particular thing is development of herd mania. Now, coming to the development of herd mania. Now, herd mania actually, you all know it is very, very important organism. It has been described by Sebastian and Das. So the development of herd mania, in this actually, it involves a free swimming tadpole larva. So the characteristic feature in the life cycle of herd mania is nothing but it exhibits retrogressive metamorphosis. Now, what do you mean by retrogressive metamorphosis? Retrogressive metamorphosis is nothing but the active larval form will actually form a sedentary adult. So that is what is called as nothing but the retrogressive metamorphosis. Now we will see how actually this active larval stage of the herd mania turns into a sedentary adult stage. Now coming to the development, of herd mania, they say that actually it has been described by the scientist called as Sebastian and Das. Now development of this particular organism is indirect because it involves a free swimming tadpole larva. And you all know actually herd mania is called as a tunicate because of the presence of the tunic or the test. So it is one of the best example of the retrogressive metamorphosis. Now, where in the retrogressive metamorphosis, already I have told you that a highly developed tadpole larva will undergo the retrogressive changes to become the most degenerated and the sedentary adult. Because when actually the development is taking place, the larva will try to gain all the caudate characters. But when once it changes from the larval to the adult stage, it loses all those caudate characters, and that means the organs they will become degenerated, and it will try to become sedentary. So you all know actually herd mania. I told you it is a sedentary animal because it attaches to a substratum with the help of the foot. So here in this case, why we call it as retrogressive metamorphosis is the tadpole larva that is developed is highly developed because it attains all the the characteristic features of the chordates. But when it metamorphoses, when it transforms from the larval stage to the adult stage, it will lose all those things. That means all the organs actually they get degenerated and it will end up in the sedentary adult. So this phenomena of retrogressive metamorphosis is peculiar to urochordates. It is peculiar to the urochordates. Okay, so that is what is called as nothing but the retrogressive metamorphosis. Now we'll see what are the gametes and the ova, how they are actually being produced and how the fertilization will take place and how actually the herd mania is being formed. Now coming to the gametes, you all know there are two types of gametes. That is the male gametes, you call it as the spermatozoa, and the female gametes, you call it as the ova. Now the sperm of herd mania actually is a flagellated structure, and it is about 4 micrometer in length, and each sperm has an anterior head which bears a middle nucleus and has a beak-like acrosome, a middle piece with a neck and a long tail. Now, according to Das, they say that sperms of herd mania are polymorphic. That means 3 types of sperms actually having acrosome, either shorter, equal, or longer than the head. So they say that actually the sperms exhibits polymorphic form. That means they exist in 3 different forms. What are those 3 different forms of a sperm? They're saying that you all know actually the sperm has the structure of, they have a head, the middle piece, and a tail, they say. So the head actually bears the nucleus, and you have a beak-like acrosome in that, and a middle piece is there, and a neck is there. So here the head bears the acrosome. So in this case, 3 types of sperms are having the acrosome. So the acrosome may be either short in size, or they may be equal in size, or they have a longer, they may be longer than the head. So they actually, when you compare it with the head, these sperms bears the acrosome, either they may be shorter, or either they may be equal, or either they may be long. So that is a characteristic feature. Now coming to the ova or the unfertilized sex, they are microlecithal. They have distinct animal pole as well as a vegetal pole. And the mature ovum is about only 0.3 mm in diameter, containing a eccentric nucleus with a nucleolus. And the egg is surrounded by an inner vitelline membrane, which is being secreted by the ovum. And outer to it, you have 2 chorionic membranes, that is inner membrane as well as outer membrane, which are secreted by the follicular cells. Now, the space between the inner chorion and the vitelline membrane actually is called as a perivitelline space, which is filled with a fluid which is called as perivitelline fluid. And this space, that is a perivitelline space, contains a number of small sized follicle cells or the test cells. Some of them actually they will float in the liquid, that is they will swim in the liquid, that is the fluid, perivitelline fluid, and most of them actually are attached to the membrane. Now these cells, what do? They nourish the ovum and they secrete a chorion digestive enzyme to facilitate the hatching. Now the follicle cells are also found attached to the outer surface of the chorionic membrane. Now, these cells will keep the ovum floating in outer perivitelline fluid. So, maturation of the ovum will occur in the sea water. Now, next is fertilization. Now, we have seen the gametes, two types of gametes, that is sperms as well as the ova. We have seen the structure, how the sperm looks like and how the ova is. Now, when these gametes are being fully developed, then the fertilization will take place. Now, in case of herdmania, the fertilization is external and cross-fertilization will take place. The spermatozoa will enter or it will penetrate the ovum near the vegetal pole. And polyspermy is not allowed by the chorionic layer. Polyspermy means many sperms. So actually, only one sperm will be allowed to enter the egg, as you all know. So here in this case of herdmania, polyspermy is not allowed by the chorionic layers. After fertilization, a number of cytoplasmic regions, they are rearranged by a process known as ooplasmic segregation. That is, yellow and cleared cytoplasm will segregate the lower hemisphere with a cap of clear cytoplasm over it, and gray yolk laden endoplasm will occupy the upper two-thirds of the egg. So that is also when once the fertilization is being taken place. So fertilization, you all know, when the gametes mature, that is both the spermatozoa and the ova mature, they come close together, okay, and they fuse and they form a zygote, you all know, okay. So, but here in this case, the fertilization is not internal, it is external and it is cross-fertilization. And the spermatozoa always try to penetrate the egg to the vegetal pole, and only one sperm is allowed to enter. Polyspermy is not allowed means only one sperm is allowed to enter the egg. Then next, when once it enters egg, then what happens? The changes will take place. The entry of the sperm into the ovum will result in the movements of the ovum cytoplasm, that is the ooplasm, so that the fertilized egg or the zygote is being formed and it is mosaic. That is, it shows presumptive areas with predictable future. So when the entry of the sperm will result in what? The ovum ooplasm will try to move apart. So when once it moves, what happens? The egg, they form a fertilized egg, they fuse and they form an egg that we call it as a fertilized egg or the zygote. And here the zygote is mosaic. That means it shows a number of presumptive areas which will form the future organs. Now, embryonic or the pre-larval development will take place. So when once that is fully developed, then the next stage of development will take place. So what is that? Embryonic development. The embryo will try to develop further. So first what happens when once the embryo is fully developed, that is the zygote is fully developed, it will undergo cleavage, it will undergo blastulation, gastrulation and all those things. Now the first is the cleavage. Now here in this case, the cleavage is determinate. The cleavage is determinate and the zygote will undergo holoblastic cleavage. Now somewhat unequal and determinate cleavage. So here holoblastic means complete and it is somewhat unequal and determinate. So that is the fate of the blastomeres is predetermined. Means here when once the zygote undergoes cleavage, the blastula will be formed and a number of cells will be developed. Those cells we call it as nothing but the blastomeres. So these blastomeres will try to develop the future the organs or the future cells or the future organ system. So here what they mean to say here is the fate of blastomere is predetermined, means before only actually it will be assigned that this particular blastomere will form this particular, this particular area of the blastomere will form this organ, this area will form this organ. So like that. So that is what is called as the fate. The fate of blastomere will be predetermined. Now the first cleavage is always vertical, meridional, and it divides the gray crescent into two unequal parts. Whereas the second cleavage is also vertical, but it is right angles to the first one. So now when once the two, the cleavages occur, it will result in 4 blastomeres. It will result in 4 blastomeres, and these 4 blastomeres, they are different. That means 2 smaller blastomeres will be at the posterior side and 2 will be larger blastomeres and they will be at the front side. So only 2 of these 4 will contain the parts of the gray crescent. Okay, so that is how the cleavage will take place. Now, these 2 cleavages will be followed by the third cleavage. So the third cleavage is called as what? Horizontal. And it passes a little above the equator and it divides the blastomeres into 8 cells, which are arranged in 2 tiers. That is, they are arranged in 2 rows. Then this is about the third cleavage. Followed by the third cleavage, you have a fourth cleavage. Fourth cleavage will result into 16 blastomeres. And subsequently, the cleavage, what it will do, it will become irregular. And finally, it will result in a solid ball of cells, which we call it as the morula. Okay, so when once a morula is developed, a single layered flat coeloblaster will be formed with an internal fluid-filled cavity, which is called as the blastocele. And it is for actually this blastocele is formed at a 64 cell stage. So in all, it takes about 110 minutes. 110 minutes means 60. That means almost nearer to 2 hours. So within 2 hours, actually, the complete cleavage will take place. So thus, a round 64 cells compact blastula is being formed containing a blastocele in the interior. The upper smaller cells are called as the micromeres or the ectoderm, and the lower larger cells, they are called as the macromeres or the endoderm. So that is how the pre-larval development or the embryonic development occurs in case of herdmania. So when once the sperm and the egg fuse, what happens? They will fertilize. When once they fertilize, it will result in a zygote. And this zygote, it will undergo cleavage. So it undergoes how many cleavages? It undergoes about 4 cleavages. So the first cleavage actually will be determinate, and it is holoblastic, and it is determinate. So it is vertical. So this will result actually in the 2 cells. Then the second cleavage actually will again is also vertical. It will again form 4 cells, means 2 plus 2, 4. Then third cleavage will be formed, which is horizontal, and it will result in 8 cells. Then the fourth cleavage will result in 16 cells. And then likewise, subsequently, the cleavage will become irregular and they go on dividing. So 16 cells will give rise to 32 cells. Then 32 cells will give rise to the 64 cells. So when once the 64 cells are being attained, what happens? A cavity will be developed. That cavity you call it as nothing but a blastocele cavity. And this compact round ball 64 cell structure, you call it as nothing but the morula. And it consists of the upper smaller cells. So this ball-like structure, on the upper side, you have smaller cells, which are called as micromeres or the ectoderm. And on the lower side, you have larger cells, which are called as macromeres or the endoderm. So that is how the embryonic development will take place. Now, next is what? Gastrulation. Now, coming to gastrulation. So now what happens? First, the zygote will undergo cleavage. So it will form the morula or the blastula, you can say. Then blastulation is followed by gastrulation. Now, it takes place by means of emboli or invagination of macromeres, which now enclose a cavity or an opening, we call it as nothing but archenteron. The cavity is actually called as the archenteron. Now, the wide ventral opening of the archenteron, you call it as nothing but the blastopore. And the archenteron will increase in size, obliterating the blastocele. That means it will try to disturb or object the blastocele. So gastrulation actually will occur by the phenomenon called as emboli or invagination of the macromeres. And it develops a cavity which is called as the archenteron cavity. And it has an opening, the opening of the wide ventral opening of the archenteron, you call it as nothing but a blastopore. Now, when once the gastrulation is being completed, the blastopore will close. That is, the opening of the archenteron will close, the blastopore will close, and the embryo begins to lengthen along the embryonic axis. So it tries to increase in length along the embryonic axis. The upper surface or the neural plate of the elongating gastrula, it will invaginate along the midline to form a groove, which will subsequently form the neural tube. And this stage, you call it as nothing but the neurula. So that is how the gastrulation will take place. Now, next is, so when once the neural tube or the neural occurs, the anterior portion of the neural tube will remain wider and will develop into a brain vesicle, and the posterior narrow portion of the neural tube will form the spinal cord. So whenever you see the development of any organism, first the brain will develop. So first the formation of the neural tube or the neurulation will take place. So that means formation of the neural tube will occur. So once the brain and the spinal cord will develop, then the hinder part of the neurula, what it will try to do, it will try to extend and it will form the other organs. Remember that, okay? So in any organism, if you take, so now the anterior portion of the neural tube, it will become wider and it will develop into the brain, and the posterior portion will form the spinal cord, Then the hinder part of the neurula, it will become extended into the tail, into which the neural tube, what it will do, it will try to elongate. So into a tail, it will become extended into a tail, into which the neural tube, the notochord, mesoderm, as well as the mesenchyme are also being extended. So with further development, what happens? the neurula will develop into an ascidian tadpole larva. It will develop into an ascidian tadpole larva. So when once the tadpole larva develops, it will rupture the egg membrane and subsequently it becomes free swimming. So that is how the larva will be formed. So next is the larval development. Now coming to the larval development, now the blastopore will close and it will develop a rudiment of tail. Again, the embryo will elongate and it will form a tailed larva. The larva actually here, it has got a tail, so it is called as a tail larva. So the presumptive notochordal cells which are there, they are being separated from the roof of the archenteron and occupies the central core of the larval tail. Then later, the archenteron will produce the presumptive mesoderm as a solid band and not as a hollow coelomic sac. Now, after 8 hours after fertilization, the chorions will rupture by an enzyme secreted by the inner follicular cells, and the fully formed larva hatches out to become the free swimming larva. So that is how actually the larval form. So this is the structure of a mature ovum. Now here they are showing. So when once the larva is developed, now the larva actually has got a tail. It is basically called as or commonly called as a tadpole larva. Now, the fully tadpole larva or the fully developed ascidian tadpole larva is very, very minute, about 1.5 mm long, and it is motile. So, its body actually is covered by a thin tunic or the test, which is secreted by the ectoderm, and it has a anterior short oval head or the trunk and a posterior long tail it has. So, that is how it will develop. We'll see now what are the various parts of the tadpole larva. Now, the first is the head or the trunk. So, at the anterior end of the trunk, there are 3 adhesive papillae made of ectoderm. Then the nervous system will be developed. The nervous system consists of an anterior enlarged hollow sense vesicle or the brain vesicle. Then the sense vesicle is elongated posteriorly into a solid thick mass of nerve cells. That is a visceral ganglion, which is again continued posteriorly into a nerve cord, which is hollow and continued into the tail. That is how the notochord will be developed. Then the sense vesicle, again, it also contains a pigmented statocyst, that is the otocyst, and 2 pigmented unequal ocelli or the simple eyes as the sense organ. So here see, this is actually the tadpole larva in the tadpole larva of Herdmania. So where you are seeing almost all the parts. Okay. The next, so when once the brain is being developed, the alimentary canal tries to be, begin, or it tries to grow from the anterodorsal mouth, leading into the narrow branchial siphon, a large sac-like pharynx. Then it will followed by short esophagus, stomach, intestine, and a small rectum. Then lastly, the rectum will end up in the anus, and the anus opens into the left side of the atrium. And then the pharynx also has a ventral endostyle and also a pair of large gill slits or the stigmata. Then again, each gill slit again will split up to form 6 stigmata on either lateral side. Atrial cavity is also formed around the pharynx, laterally and dorsally, and opens to the exterior by means of atrial aperture. Then the heart and the pericardium is also formed, and it is located postroventrally in between pharynx and the stomach. The mesenchyme cells found scattered all over the body beneath the ectoderm, and it lies in the posterior region of the trunk. So that is how actually the larva is being formed. Next is the tail. Now the tail is also a very powerful organ being laterally compressed, and it is pointed terminally, and this tail actually is long, 0.9 mm long, and it is fringed with a vertical continuous tail fin formed by the test. You see here, this is the tail, you have got the tail fin. This is how it is developed and marked with oblique striations looking like the fin rays. So it contains a dorsal tubular nerve cord, a notochord beneath the nerve cord, and lateral muscle bands, just like the adult chordate features. So the anterior end of the notochord will slightly project into the trunk region. Then the ascidian tadpole larva actually is a true chordate. Why we are calling it as a true chordate? I have told you, because when the Herdmania is being developed at the larval stage, it actually attains all the chordate structures like the presence of a notochord, then dorsal hollow tubular nerve cord, and also the pharyngeal gill slits. But when once this tadpole larva gets transformed from the larval stage to the adult stage, all these organs, they get degenerated and it will become sedentary. It will become sedentary. So that is why that phenomena you call it as retrogressive metamorphosis. It is called as retrogressive metamorphosis. So next, the anterior end of the notochord will project into the trunk region. So the ascidian tadpole larva actually is a true chordate. So having a dorsal hollow neural tube, a notochord in the tail, and also paired gill slits in the pharynx. And after about 3 hours of active non-feeding and free swimming existence, it sinks to the bottom. It will sink to the bottom. So when once bottom means bottom of the water. So it will sink to the bottom and later on it will develop into the adult. So this is about the tadpole larva of Herdmania. So now we'll see how actually this larva will exist and what is its habitat, where actually it will try to live. So just after hatching, the larva actually will become it is photopositive and it is geonegative. Photopositive means it is attracted towards the light. So it is light positive, you can say, and geonegative means towards the earth, gravitational earth. So it is geonegative. So it cannot feed because its mouth is still closed by the test. And after a short active free swimming existence for about 3 to 4 hours, the larva will become geopositive. Once it becomes geopositive, it will, what happens? It will go towards deeper and it will become photonegative that time, and it will become sluggish. It will become photonegative and it will become sluggish. Then it sinks to the bottom, attaches itself upside down to a hard substratum by means of an adhesive papillae, and it will undergo rapid degeneration or retrogressive metamorphosis to turn into the adulthood or to attain adulthood. So according to the Beryl, the selection of a suitable habitat is essential as a larva may not survive or any other habitat or may get suffocated by the bottom mud and the detritus. So it has to, when it is, when it has attained or when it is about to attain the adult stage, it need to first select a substratum, a solid substratum where it can survive. So that is how actually the tadpole larva of the Herdmania will get transformed into an adult. So basically they say during larval stage, it has all the chordate structures. But when once it gets it start slowly, it will all those what do you say, the important features like presence of a notochord, the pharyngeal gill slits, as well as nerve cord, they will get degenerated. They will get degenerated. So initially it is photopositive and geonegative. So after 3 to 4 hours, what happens? It will become photonegative but geopositive. Okay, and it will become slowly, the organs also will get these structures will get degenerated and it will become sluggish and sink to the bottom. And it tries to when once all the structures get degenerated, it will sink to the bottom and it will try to get attached to the substratum and it will try to attain the once it attains the adulthood, then it will lead a sedentary life. So that is about the what do you say, the development of Herdmania. Okay, is it okay, students? Hello, hello, hello. Okay. So with this, we have completed the type study, that is Herdmania. So this Herdmania, it comes under the Eurochordates. You all know actually protochordates. I have already in the classification I have told you protochordates have been divided into 3 classes, that is Eurochordates, cephalochordate, and hemichordates. So in this you have got one example that is called as Herdmania, which is commonly called as the sea skirt. And I have already discussed about its external features, the external morphology, body wall, digestive system, then circulatory system, respiratory system, everything we have completed. And today, with this reproduction, we have completed the whole. Now, next chapter we'll go, that is Amphioxus. We'll go to the Amphioxus. You've been, hello students, you be online. I will just try to share the other notes that is on Amphioxus. Hello. Now we will start with the Amphioxus, which is also called as branchiostoma. Now, coming to Amphioxus, now this is another type study which is given in your syllabus. Now, coming to the Amphioxus, you all know it is basically called as branchiostoma. It has been, identified, you can say, or it has been described or classified by the scientist called as Pallas, P.S. Pallas, in the year 1974. And he named this particular organism as Limax lanceolatus. He called it as Limax lanceolatus and described it as a slug. But he had ill-preserved the specimens on which he worked. So therefore, this name was rejected. So later on, in the year 1836, the scientist by name Yarrell recognized the specific nature of this particular animal, and he called it as Amphioxus lanceolatus. So initially it was called as Limax lanceolatus by Pallas, but it was called as Amphioxus lanceolatus by the scientist called as Yarrell. He discovered that later on it was discovered that O.G. Costa had christened them as branchiostoma. So what he did is, actually after Amphioxus lanceolatus, the scientist by named Costa, he called it as branchiostoma in the year 1834. So like that, the name of this particular organism changed from first Limax lanceolatus, later on as Amphioxus lanceolatus, and later Costa called it as branchiostoma. Now, according to the present, the study or the present situation, now we call this particular organism either as Amphioxus or either as a branchiostoma. So this particular, the names given by both these scientists actually are now being uniformly accepted by almost all the other scientists. So therefore, the name branchiostoma is accepted as the generic name, and we use the Amphioxus as a common name. So we can call it as the branchiostoma as a generic name and the Amphioxus as a common name. So we can say branchiostoma as a generic name, that is the genus, and the Amphioxus as the common name. So this is about the brief history of branchiostoma. Now coming to the distribution, now where actually this particular organism is being distributed, where it is located. Now they say that actually the Amphioxus has a cosmopolitan distribution. Cosmopolitan distribution means it is present almost throughout the world and found only on the sandy shores covering the tidal area to depths and many meters. So it is an inhabitant of the tropical as well as the temperate seas. And the most common lancelet, that is branchiostoma lanceolatus, or you can call it as the Amphioxus lanceolatus, whatever you want to call it. So it has been recorded from the west and southern European coast, on the East African, and also from the western and the southeastern Indian coast. So that is what. So geographical distribution means it is cosmopolitan, almost found throughout the world. But the common lancelet, which is basically recorded from the west and south European coast and also East African coast, is called as branchiostoma lanceolatus. Now we'll see the about the branchiostoma lanceolatus, where actually it is being found. Now branchiostoma actually lives in both marine as well as the euxerine habitat. It is commonly found in the sandy shores, wherever you have the sand. So in that particular area, you find this particular organism in more number. It lives in dilute seawater, having the 15 to 33 salinities, and it is mostly marine in its distribution and stenohaline. Stenohaline means actually it is restricted to a narrow range of environmental salt concentrations. Means it cannot, try to overcome the salt concentration. So that is the reason why it is restricted only to a narrow range of environmental salt conditions in its behavior. So it leads a dual life. It leads a dual life means it is both sedentary as well as the it can actively swim in water. So both it can attach to substratum, and also if time permits or if it wants, then it can also swim. So it needs a dual role. It is a sedentary animal, and also it can swim actively in water, and it swims vertically in the water. So it swims, they say, actually like a fish. So it has a fish-like appearance, they say, and the fins here do not play any role during the swimming. Whereas in fishes, the fins will play a role, but in this case, the fins will not. Though it looks like a fish, but though it has got the fins, but the fins will not help in the swimming as the case in the fishes. Now it burrows in the sand but remains always at a smaller depth. It keeps the anterior end of the body projected above the sandy bed to maintain a constant water current passing through the mouth and also and expelling it through the atriophore. So that is about the habitat of Branchiostoma. Now, it feeds on microorganisms brought into the pharyngeal cavity along the respiratory water current. So whenever actually through the mouth, so whenever it wants to, it respires along with the water current, the food particles actually will enter. So the always basically feeds on the microorganisms which are brought up along with the respiratory water current into the pharyngeal cavity. And it exhibits a ciliary mode of feeding. It exhibits a ciliary mode of feeding. So this is actually the structure. So this is how the lancelet or the amphioxus will look like, also called as branchiostoma. So this is the longitudinal section. Okay. Now coming to the other aspects now, they say branchiostoma is about 3.5 to 6 centimeter in length, and the body is very, very slender, and it is translucent. Superficially, it looks like fish and laterally compressed and pointed at both ends. Hence, it is the name lancelet. Why they have given it as lancelet is, though it is laterally compressed and but it is here. It is pointed at both the ends. So that is why it is called as the lancelet. Now, the anterior two-third of the body is roughly triangular in shape. The anterior two-third of the body is roughly triangular in shape, and the posterior third is oval in shape. And the pointed anterior end of the trunk projects in front as a snout or as a rostrum. See here. Now this is the anterior region. So it is called as a rostrum. So this is about the thing. The next is below the rostrum, you have an oral hood which is formed by the dorsal and the lateral projections of the body. So just beneath the rostrum, you have got a oral hood. And this rostrum bears 20 or more stiff oral cirri on the tentacles. So it bears, so this is the tail region, this is the posterior region, and here you have the anterior region. See, this is the buccal cirri, just now I have told you. So the rostrum actually, below the rostrum, you have an oral hood. Here, this is the oral hood, and this is the buccal cirri, this is the rostrum. And here, this is the notochord. So that is how the structure is. So the rostrum, below the rostrum, we have an oral hood, and this oral hood bears the oral cirri or the tentacles. This is the buccal cirri or the oral cirri. So this is about the external morphology. Now coming to the body. Now the body, they say, of this particular organism is divided into two regions. They are called as a trunk region, and this is called as a tail region. So from here to here, it is called as a trunk, and from here it is called as a tail. So the body is divided into two regions, the trunk region as well as the tail region. And the true head is lacking. So it doesn't have a distinct head. So that is why they say the true head is lacking. So you don't find the distinct head. So it just looks similar. So head and the trunk. So that is why they say the body is divided only in the two regions. That is the trunk and the tail. Now coming to the fins. The branchiostoma actually consists of three unpaired fins. It has got three unpaired fins, that is the dorsal fin, caudal fin, as well as the ventral fin. And the paired fins are absent. Whereas in the fishes, we see paired fins. But in case of this, of course, not all the fins in the fishes are paired. Some of them are paired and some of them are unpaired. But in this branchiostoma, you have all the three unpaired fins. Now, the paired fins are totally absent. Along the mid-dorsal side of the body, there is a dorsal fin which runs along the whole length. There is a dorsal fin which along the whole length. Now, it is joined to somewhat broader caudal fin around the tail. Now, around the caudal tail. And mid-ventrally, there is a ventral fin which runs from the caudal fin to the atriophore. And the dorsal fin is supported by a series of fin rays. So that is how. So we can say the branchiostoma has three unpaired fins. They are dorsal fin, caudal fin, as well as a ventral fin. Now, these fins possess the fin rays. And these fin rays actually are made up of gelatinous substance as well as the connective tissue. They are made up of connective tissue as well as the gelatinous substance. And the fin rays are lacking in the caudal fin. In the caudal fin, you don't find the fin rays. And the fins do not have the same structure as that of the fins of fishes. Already I have told you, even the fishes have got the fins. But though this particular organism has got the fins as that of fishes, but they are not similar to that of the fins which are found in the fishes. So next is the folds. Now they say the oral hood actually is continuous posteriorly The oral hood The oral hood is continuous posteriorly with two lateral membranous hollow metaplural folds running ventrally up to the atriophore. So the oral hood actually is continuous posteriorly with two lateral membranes, and both the folds actually are joined by a horizontal fold of body wall called as the epifloor, and it forms the floor of the atrium. So apart from the fins, it also has a oral hood, and this oral hood actually, the oral hood, it has got the folds. The folds are nothing but the the membranous structures of the oral hood. So we can say the oral hood continues posteriorly into two lateral membranes, which are called as nothing but the metapleural folds. Now next is myotomes and gonads. Apart from the fins, folds, it also consists of the myotomes as well as the gonads. So the body wall of this particular organism shows metameric segmentation, that is having the metameres being made up of made up of the number of muscles, which are called as the myotomes or the myomeres. So they also possess the myotomes. A number of blocks of muscles are there, which are called as the myotomes or the myomeres. Now these are found just beneath the body wall. They are found beneath the body wall, and beneath these myotomes on both the sides, you have a series of gonads are present, which occurs between the mouth as well as the atriophore. So we can say branchiostoma possesses the fins, it possesses the folds, it also has the muscles which are called as the myotomes, and it also has the gonads. The next, apart from this, you also have the apertures. Now below the pointed anterior extremity is a large median aperture, the mouth being surrounded by a frill-like membrane, the oral hood. That is called as the oral hood, and its edge is beset with numerous tentacles or the cirri. Then a short distance from the posterior extremity of the body, that is a trunk, you have the anus, which is actually placed on the left side at the base of the caudal fin. And the post-anal portion of the body is called as nothing but the tail. Then this is all about the external morphology. Now coming to the musculature, that is the muscles. Just now we have seen it has got the myotomes. So myotomes are nothing but the muscles. So here musculature. So the muscles which are present beneath the body wall, they exhibit metameric segmentation. They exhibit metameric segmentation, and they consist of a large number of muscle segments, which are called as the myomeres or the myotomes. About 60 to 62 muscle segments or the myomeres are present, which are being separated from one another by means of partitions. And these partitions are nothing but made up of dense connective tissue called as the myocomas. They are called as dense connective tissue called as the myocomas. And they have the appearance in the form of V. They occur in the form of a V shape with their apical portion directed forward. And the myotomes also have striated muscle fibers which lie longitudinally. They are inserted at each end into the myocomata. And these, the contractions of these myotomes will cause side-to-side bending of the body. And the myotomes of the two sides, they do not correspond, but they are placed alternatively. So this is the musculature, or you can say about the muscles. Okay. Then next is you have got the nerve cord as well as a notochord. Now they also have a nerve cord as well as a notochord, and this is a characteristic feature of the vertebrate, as I told you. But in many invertebrates, the muscles are of uniform thickness all around. So ventrally between the metapleural folds, in the floor of the atrial cavity, you have a special set of transverse muscles in the anterior two-thirds of the body. So no such muscles are seen in the vertebrates. Are seen in the vertebrates. So you although actually it possesses the characteristic feature, some of the characteristic feature of the invertebrates also. So we can say just it is a connecting link between. So it has got the affinities which similar to that of the invertebrates also. So that is why it exhibits the similarity. So it shows affinities to both vertebrates as well as the invertebrates. So that is why it is not included completely in the invertebrata, nor it is included completely in the vertebrata. You all know actually protochords. So protochordates basically actually are having the features of both the invertebrates as well as the vertebrates. So that is why this particular the subphylum has been excluded from the invertebrates to that of the vertebrates. You all know that. Okay. So this is actually the branchiostoma to show the internal anatomy. So just now we have seen how actually it looks like. So this is about the digestive system and all. Okay. Now we will see about the different systems now. So this is only an external morphology I have told you, that is about the amphioxus, which is also called, you can also call it as branchiostoma or branchiostoma, or the lancelet. It is also called as a lancelet or the amphioxus. So branchiostoma, generic name is the branchiostoma, and the specific name is the lancelet. So this is all about the branchiostoma, that is amphioxus. We have seen where it is being formed. We know it is cosmopolitan in distribution, means found throughout the world. It lives both in the marine as well as the issuerent bodies. It is commonly found in the sandy shores. So it possesses the fins. It just looks like a fish. Okay, with the laterally compressed body, it has got the fins, as I told you. So now it is divided into, the body is divided into the trunk as well as the tail. The distinct head is not there. It lacks the distinct head. So that is why it is divided into trunk and tail. It possesses the fins. Then it has got the folds, myotomes, as well as the gonads. Myotomes are nothing but the muscles, which are called as also the myomeres. 62 myomeres are present. And these myomeres actually are being separated from each other by means of a dense connective tissue, which are called as myocomas. So this is how the structure looks like. Now we'll go to the other systems. Okay, students, now we will go with the digestive system of amphioxus. Now you all know the digestive system is nothing but the alimentary canal. It consists of the alimentary canal as well as the digestive glands. Now, the alimentary canal of this amphioxus is a very straight tube running from the mouth to the anus. Now, the different parts, as you all know, of the alimentary canal are the mouth, the oral hood, and the buccal cavity. Now, the first is a mouth. The mouth actually is a very, very small rounded aperture. It is situated at the base of the vestibule or the buccal cavity of the oral hood. It is surrounded by a membrane, which is called as velum, and it acts as a sphincter bearing 12 slender velar tentacles or the languettes on its free edge, and it helps to close or open the mouth. So this is the first part of the alimentary canal. So what is the first part of the alimentary canal? The digestive system, that is nothing but a mouth, which is a small rounded structure present at the base of the vestibule or the buccal cavity of the oral hood. It is surrounded by a membrane called velum. This velum will act as a sphincter. Sphincter is nothing but an opening, and this bears 12 slender velar tentacles. These tentacles are also called as languettes on its free edge, and it helps to close or open the mouth. Now, the mouth is being followed by oral hood and the buccal cavity. A large median aperture is situated just beneath the tip of the anterior end. The tip of the anterior end you call it as the rostrum. So this aperture is surrounded by a frill-like membrane, which is called as nothing but the oral hood. Now, the membranous lateral and the ventral margins of the oral hood, they are fringed and they bear the buccal cirri. Already we have seen in the diagram, okay, that buccal cirri. And this buccal cirri actually consists of a number of sensory cells, some of which of them are called as mechanoreceptors. They are called as a mechanoreceptors, and these sensory cells actually provide the animal with the information in the water current that is going on. Now, the oral hood as well as the buccal cirri, they are supported by the skeletal rods. And during the inflow of this water current, the buccal cirri will form a sieve to prevent the entry of the large particles. So, the presence of buccal cirri in case of amphioxus is very, very important, because whenever the water current, along with the water current, the food enters into the pharyngeal cavity. Now, this buccal cirri, what it will try to do, it will try to sieve. It will sieve in the sense, it will try to allow, prevent the entry of larger particles. Only the smaller particles will be allowed to enter into the pharyngeal cavity, but the buccal cirri will not allow the entry of the larger particles. So that is a characteristic feature of the buccal cirri. Then The oral hood will enclose a cavity which is called as the vestibule or the buccal cavity. Now, this buccal cavity actually is a short part of the foregut. It will receive the water as well as the food particles from the mouth. So first we can say the first part is the mouth, followed by the oral hood and the buccal cavity. Then apart from this, you also have a veal organ. Now we'll see what is veal organ. Now the inner lining of the vestibule, it produces or consists of a complicated ciliated groove and ridges called as the veal organ or the rotatory organ or the ciliated organ. Now this has been named by Muller. So that is why it is called as a ciliated organ or a veal organ of Muller. Now this veal organ produces a whirling current of water. It produces a whirling current of water to sweep the food matters into the mouth. So the presence of this veal organ will allow, or it will try to actually sweep the food particles along with the water and allow it to enter into the mouth. So that is actually the main function of this particular veal organ. Then next you have Hatch's groove. Now Hatch's groove is nothing but it is a sedated glandular groove which runs along the roof of the vestibule, or the buccal cavity. Now this is being lined partly by the endocrine cells which are believed to secrete the pituitary-like hormones into the blood. And the groove of this Hatch's, it will terminate into a small depression which is called as the pit of Hatch's, and the left embryonic coelomic cavity of the region will become reduced to form the pit. So Hatch's groove and as well as the veal organ actually have got their important role to play. The next is what? Velum. Now there is a ring of finger-like sensory tentacles around the mouth which is called as the velum. Already we have seen the velum. So this will separate the vestibule, that is the buccal cavity, from the pharynx. So we can say the structures are: first is the mouth, followed by the oral hood and the buccal cavity. And before the buccal cavity actually joins the pharynx, you have got a veal organ, then Hatch's groove is present and followed by the velum. The next is the pharynx or the bronchial sac. Now the pharynx is a large laterally compressed tube which will occupy more than half of the total surface area of the body. It occupies total surface area of the body. Then the lateral wall of the pharynx is perforated obliquely with arranged vertical apertures called as the gill slits or the bronchial slits. And they say about 180 pairs of gill slits are being present, and they open into a U-shaped special cavity which is called as the atrial cavity or the atrium or the peribronchial cavity, which surrounds the pharynx on all the sides except the dorsal side. And this atrium is closed anteriorly, but it opens to the exterior to an aperture which is called as atriopore, which is just situated behind the level of the pharynx. And the whole cavity of the atrium is lined by the ectodermal epithelium of the ectodermal origin. So that is about the pharyngeal region. Then next you have the endostyle. The endostyle actually is nothing but a ciliary and a glandular groove which is present on the floor of the pharynx. It is present on the floor of the pharynx, and it is the histological structure of this is almost similar to that of the bronchostoma. In bronchostoma also is similar to that of ascidia, that is Herdmania. So they say that actually the endostyle of bronchostoma is almost similar to the endostyle that is being found in the ascidia. That is histologically they are almost similar. So now the pharynx will lead into the endostyle. Now next is what? The epipharyngeal groove. Now the epipharyngeal groove is again a ciliated median groove which is present on the dorsal side of the pharyngeal cavity, and it is also called as epipharyngeal cavity or the hyperpharyngeal groove. And this epipharyngeal groove will join with the anterior end of endostyle by peripharyngeal ciliated tract. Now next is your esophagus. So you all know actually first is the mouth, then you have the oral hood and the buccal cavity, then the buccal cavity is being followed by the pharynx, and pharynx is being followed by the esophagus. In between, the other structures are being present, like you have the velum, then you have got the endostyle and all those things. Okay. Now next is esophagus. The pharynx will lead into a short, narrow tube which is ciliated, called as the esophagus. This esophagus again will open into the midgut, and the pharynx as well as the esophagus both constitute the foregut. So we can say pharynx as well as the esophagus will form the foregut, and the foregut will lead into the midgut. And our midgut actually consists of, you all know, the intestine. So the intestine, that is the gut region, the major gut region, is a straight tube. It is ciliated, which is divided into two regions, that is called as the midgut as well as the hindgut. So here they say the pharynx as well as the esophagus, they form the foregut. The foregut will lead into the midgut as well as the hindgut. So the intestine actually is divided into two regions, that is the midgut as well as the hindgut. Now midgut will include the hepatic diverticulum and the ileocolonic ring. And at the junction of esophagus and the midgut, then there is a large single unbranched out pouching lying at the right side of the pharynx called hepatic diverticulum. And the posterior part of this hepatic diverticulum, the midgut will narrow into a short, ciliated, wider section, which is called as the ileocolonic ring. Then the tract of the intestine is provided with a weaker musculature. So that is how the intestine is. Then this intestine will end up in the anus. It will proceed posteriorly as a single straight hindgut, which will open to the exterior by means of an anus. The next, this is how the digestive system is. So the digestive system of the branchiostoma. So here you have the mouth, then you have the buccal cavity for a buccal cirri. So in this region only you find the bile organ, you have got the velum, then you have got this buccal cavity will lead into the pharynx. The pharynx will have the gill slits, or also called as the pharyngeal gill slits. And this pharynx and esophagus, pharynx will lead into esophagus. And here you have a hepatic diverticulum. Both pharynx and esophagus, they form the foregut, and this foregut will lead into the intestine. That is, intestine is divided into two regions, that is a midgut as well as the hindgut. Okay, and it has got an atriopore, and the intestine will finally end up with the anus. So this is how. Then apart from this, it has hepatic diverticulum. So that hepatic diverticulum, in case of this, you call it as nothing but the liver. So the so-called liver, in this case, it is referred to as digestive gland in the branchiostoma. So we don't call this digestive gland, that is the liver. So it is not called as digestive gland. It is called as the hepatic diverticulum. Now it is a large blind outpouching. Now it contains the zymogen cells. Now these zymogen cells will produce the digestive enzymes called as the lipase as well as the protease, and they are carried into the lumen of the midgut by the ciliary cavity. It is also a part where the fat is deposited. So the liver, you can say the so-called liver, which is called as a hepatic diverticulum in case of branchiostoma, is an important organ. It is a large, the blind outpouching containing zymogen cells, which produces the digestive enzymes. Then the mechanism of feeding is, you all know, actually it is a microphagous animal, and the food consists of protozoans, algae, diatoms, as well as the other organic particles. So it is microphagous, and it feeds only on the small microorganisms. So this is about the digestive system of the, the branchiostoma or the amphioxus. Now we'll go to another system, that is the circulatory system. Sorry, 1 second, students. We'll go to the nervous system. Okay, first we'll see the nervous system. Now coming to the nervous system of branchiostoma. Now the nervous system of branchiostoma is very, very simple compared to vertebrates. It consists of a dorsal nerve cord and a nerve net. So the dorsal nerve cord actually runs along the length. See, this is the nervous system, the anterior part of the nerve cord in the dorsal view. So it consists of a dorsal nerve cord as well as a nerve net, which is this dorsal nerve cord as well as the nerve ring. It runs along the length of the body and it is situated just above the notochord. And the nerve cord actually is considered to be homologous to the spinal cord of vertebrates. So it is almost similar to that of the vertebrate spinal cord. It is a primary center for coordinating the responses to the sensory stimuli and also controls various activities. Now next is the nerve net. Now the nerve net consists of a diffuse network of nerve cells. It consists of a diffuse network of nerve cells spread throughout the body. It serves primarily to transmit the signals and also coordinate the functions. So the nerve cord, the central nervous system of the branchiostoma includes a hollow dorsal neural tube or the nerve cord lying mid-ventrally just above the notochord. It has no ganglia. It terminates in the rostrum just behind the anterior end of the notochord. It shows a little enlargement, the so-called brain or the cerebral vesicle. Then the posterior part is called as the spinal cord. This spinal cord will gradually taper to the end just before the posterior end of the notochord. Apart from this, a narrow central canal called as a neurocele will run throughout the length of the neural tube, and it is filled with a fluid which we call it as the cerebrospinal fluid. Now, it dilates with the cerebral vesicle, forming the ventricle. From the roof, there is a pouch-like, arises a pouch-like structure, a pouch-like blind dorsal diverticulum, which extends behind over the central canal for a shorter distance. And the central vesicle contains two important organs. They are a pigment spot in the anterior wall and an infundibular organ on its floor. Then histologically, when we see, the nerve cord resembles that of the vertebrates. So it consists of an inner gray matter of nerve cells surrounding the central canal and the outer white matter of the nerve fibers. That is how the nervous system of the branchiostoma looks like. So this is the VLS of the anterior part of the nerve cord. Then coming to the nerves, the peripheral nervous system, you all know actually the central nervous system consists of the brain as well as the spinal cord. Now coming to, and the peripheral nervous system consists of the nerves. So the peripheral nervous system, we will include the paired nerves which arise from the nerve cord. The first two pairs arise from the cephalic vesicle. They are called cerebral nerves. They lack the ventral roots and they are purely sensory in nature, and they convey the impulses from the receptors of the snout, oral hood, and buccal cirri to the nerve cord. The nerves that arise from the nerve cord behind the cerebral vesicle, they are called as the spinal nerves. So one pair of these spinal nerves arises on either side in each segment, and each pair of spinal nerve will actually include separate dorsal as well as the ventral nerve roots, which do not unite to form a single mixed spinal nerve as you see in the vertebrates. Dorsal nerve root is both sensory and motor and passes out to the skin between the myotomes. The ventral nerve root is motor and it arises opposite to the myotome. So thus the dorsal as well as the ventral root of the pair, they do not originate at the same level. But the dorsal root of one side will lie opposite to the ventral root of the other side. So that is how actually they arise. While the dorsal root is single, but the ventral root actually is made up of several branches. And the nerves of this amphioxus, they are all very, very primitive and they are non-myelinated. That means they do not possess the myelin sheath. They are non-myelinated and they are not covered by a sheath of myelin as we see in the vertebrates. Then coming to the autonomic nervous system. Autonomic nervous system consists of two nerve plexus which are present in the gut wall. Then next you have two nerve plexus are there in the smooth muscles of the wall of the intestine, which are connected to the neural tube by the visceral nerves. And the autonomic system, it controls the involuntary muscles. So that is involuntary muscles of the gut wall. So this is about the simple, the basic plan or the structure of the nervous system of the amphioxus. Okay, students. So with this, now for today, we will close the class. And the next week, we will continue with the other chapters.